I hope that you can hear and see me. So I'm Athena Akrami, a neuroscientist at Science Based Wellcome Center at UCL. Today we have 3 exciting talks. Before that, I would like to have a couple of announcements. One is for poster presenters. So you might know that there is a personal chat attached to your poster booth in the Hopin. And so you can see the questions that people put there on your poster, and then, but you cannot see your presentation. So, so we encourage you to answer these questions in the same chat window. Okay, so this is for poster presenters. Then speakers, there is a session option in Hopin where you can create your own session where participants can join and continue discussion with you after your talk. So please let the audience know if you intend to create this session and when you will be available to have this, the Q&A with them after your talk. And both the poster booth and the session bottoms are at the left-hand side of the screen in Hopin, okay? And those of you who have signed up for Cosyne in the last 24 hours, please note that it takes about an hour to gain access to the Gather Town after you have registered. So be patient, and because access has to be granted by hand, and our tech staff, they are super busy. Please be patient, and within an hour you should have access to the Gather Town. Okay, so I think we can have now our session start with our first speaker, Jae-Hyun Kim. Can we have him on the stage, please? Hello, Jae.
Hi. Yeah,
hi. So, yes, we hear you. Jae-Hyun is going to present his work from the lab of Seung-Hee Lee in South Korea, and the National University in Daejeon, South Korea. And here we are. Thank you, Jae.
Thank you for introduction. So right now, can you see my screen?
Yes.
Great. So, hello everyone. I am Jaehyun Kim from KAIST. Firstly, thank you for the abstract reviewers and Cosyne organizers giving me this great opportunity to present my research. Let me start. Sensor-guided and goal-directed action is a fundamental process in our life. Based on the sensory input like traffic lights, our brain operates a series of neural processing to generate a goal-directed action, such as pressing the gas pedal. I'm interested in the neural circuit mechanism underlying this process, particularly in the higher cortical areas, such as anterior cingulate cortex. But this is still unknown, so this is my interest. So, yes, according to the recent tracing studies, mouse anterior cingulate cortex, ACC, is interconnected with visual areas and strongly innervate motor areas like the dorsomedial striatum. Furthermore, it has been suggested that the ACC collects sensory information and controls action decision in both primates and rodents. Therefore, we decided to examine ACC and how the circuits in the ACC process visual information and generate goal-directed action. To study this, we trained mice to detect visual stimuli using simple visual detection task. Here, this video clip shows whisker, nose, and licking behaviors from task-performing mouse. The video motion energy calculated from these areas show that the mouse reports licking actions and correlated orofacial movements after visual stimuli. Before task learning, novice mouse showed impulsive and useless lickings before visual cue. After task learning, Expert mouse showed much more efficient leakings after stimuli while suppressing impulsive leakings. The leaking after visual perception, quantified as perceptual leak heat trials, increased in the expert mouse, and the impulsive leaking, quantified as first alarm and impulsive leak heat trials, were reduced after task learning. To test which brain areas are required for mice to carry out this task, we inactivated V1, V2M, and ACC using Mushimo. Both V1 and V2M inactivation impaired the task performance by increasing mistrials. Interestingly, however, ACC inactivation induced a significant surge of first alarm trials and impulsive leaking heat trials. These results tell us that ACC is critical to withhold leaking actions in the absence of visual cue. Next, we moved our focus to measuring neural activities in ACC and V1 from task-performing mice using multi-channel silicon electrode. We firstly defined sensory and motor neurons according to their activity changes with stimuli and leak onset. We collected 550 ACC neurons and 175 V1 neurons from task-performing mice and classified their responsiveness. Mice showed trial-to-trial variability of leak times in perceptual heat trials. Gray lines here indicate the leak time distribution from the individual sessions. We then sorted heat trials according to the leak response times and compared neural activities in early leak to late leak heat trials. Interestingly, different types of ACC neurons showed distinct activity patterns across the trials. First, ACC sensory neurons that show increasing activity in response to the visual stimuli showed significant correlation between visual evoked activity and the leak latency. On the other hand, the ACC motor neurons that show suppression in their activity prior to the leaking showed the temporal correlation with leak response time. Other types of ACC neurons did not show any such trial-based correlation with leaking behavior. However, V1 sensory and motor neurons did not show any such correlation. So far, our data showed that ACC neurons represent the timing of a goal-directed action, that is leaking, during visual detection task. Sensory increasing signals and motor decreasing signals reliably and significantly represent leak response time. We next wondered whether the sensory increasing neurons directly suppress motor neurons to evoke leaking in task-performing mice. To examine this, we tried to label visual inputs to the ACC with channelodopsin to optogenetically activate these neurons in the ACC and measure the premotor suppression and leaking behaviors. We firstly identified the visual inputs to the ACC by injecting electrode AAV-TV tomato into ACC. We found that the V2M was the major input area projecting to the ACC among multiples of lesions in the visual cortex. We then labeled V2M recipient ACC neurons with Cre by injecting AAV1 Cre into the V1 and further with the channelodopsin by injecting AAV flex channelodopsin into the ACC. We then used optic fiber conjugated silicon probe to measure blue light driven activity in the ACC. Short pulses of blue light successfully identified channelodopsin labeled V2M recipient ACC neurons. Interestingly, among 20 optotac neurons, about half of them were fast spiking and sensory increasing neurons that show a significant correlation between the visual signals and leak response times. This proportion was much larger than the non-tech neurons, suggesting that the major recipient of V2M inputs were fast spiking and sensory increasing neurons. We next activated these neurons with the blue light that have the same temporal structure as the visual stimuli during the task. Even in the absence of visual stimuli, photostimulation evoked leaking behaviors. Both soma in the ACC and axon of the V2M neurons evoke leaking. The control EYFP expression did not induce any leaking. Furthermore, when we activated the V2M recipient neurons in the ACC, the neighboring neurons in the ACC were strongly suppressed compared to control group. Together, our data indicate that the visually evoked ACC neurons that receive inputs from the V2M causes the premotor suppression and leaking responses. The majority of V2M recipient neurons in the ACC was fast spiking neurons, which induced feedforward inhibition in the ACC to trigger leaking. This result well explained the mutual inactivation data suppression of ACC activity caused increase of impulsive leaking. One remaining question was how the spontaneous leaking was evoked even in the absence of visual stimuli during the task. We first examined visually evoked activity of sensory increasing neuron during perceptually, missed, and spontaneously. Interestingly, async neurons showed significant increase in the activity before the spontaneous leaking. Although the level of increase in the activity was mild and smaller than the visually evoked activity, it was sufficient to evoke motor suppression in AMDIC neurons. But in mistrial, even though the activity of async neurons was stronger than before the spontaneous leaking, the AMDIC suppression and leaking was not induced. Resolving this discrepancy, we found that other neurons in the ACC showed significant decrease in their activity before the leaking. This decreased activity was different from the fast and transient AMDIC signal, showing temporally longer and persistent at the network level. This network activity also correlated with leak response times in perceptual heat trials and showed a gradual drift across the session. These results suggest that the ACC network activity gates visual to motor suppression to evoke leaking during the task. Gradual drift in the network activity suggests that this activity can be modulated by internal states of the animal, such as motivation and task engagement during the task. This is the summary illustration of our data. We found that ACC neurons show different activity patterns in perceptual leaking, missed, and spontaneous leaking conditions in mice performing the visual detection task. During the perceptual and spontaneous leaking, low level of recurrent network activity in the ACC promote the sensory evoked motor suppression to release the leaking action. During mistrial, however, visual evoked activity was intermediate and the network activity was high, so the motor suppression was not induced. Our model demonstrates that the network activity of the other neurons in the ACC gates the sensory evoked motor suppression in the ACC. Finally, we wondered whether the ACC activity is involved in stopping leaking actions. To examine this, we devised a stop signal test in which we trained mice to pause leaking after stop auditory cue in a subset of visual goal trials. Mutual inactivation of ACC increased impulsive leaking as well as a decrease in stopping leaking in stop trials. When it recorded ACC neural activity during the stop signal task, we found that the ACC motor decreasing neurons now show an increase in their activity during stopping the leaking. Furthermore, ACC network activity measured in the other neurons showed a significant increase in successful stop trials. Altogether, our data demonstrate that the ACC activity gates visually guided goal-directed action in task-performing mice. The leaking decision was evoked by suppression of motor neurons in the ACC, and sensory inputs trigger the sensor-to-motor feed-forward inhibition to evoke this suppression. In mistrial and stopping the action condition, both motor neuron and network activity increased and leaking was suppressed. So this is our lab photo taken recently. I particularly appreciate my PI, Dr. Seung-Hee Lee, supervising and supporting my research, and other students, Dong-Hyun Ma, Eunji Jung, and Ilsong Choi also participate in this project. Thank you for your attention.
Thank you so much, Jae-Hyun. This was great, and you really finished it before the time was up. We have about like more than 5 minutes for questions, about 7 minutes for questions left. So, so please, everyone, put your questions on a Slido so we can have them here, and then you can vote on questions too. So we have one question already posted here. Jae-Hyun, the question is there a direct interaction between ACC and ALM, which also has been shown important in leak decision-making tasks?
Yes, that is very important question, I think. So actually, I already conducted some tracing study about that. Specifically, I injected the lateral AAV TB tomato into the ALM area, but I cannot see a very small number of ACC neurons projecting to the ALM. So a little bit. Yes, there is, but very small number of neurons projecting to the ALM. A little, very weak projection from ACC to ALM.
I see. Did you try to manipulate that projections?
Not yet. So since I only observed very small number of neurons that projecting from ACC to ALM, so I didn't try that.
And so maybe follow up to that question. So there are also, you know, so my question is that if the output of the task is different, is not leaking, it's like orientation, do you think ACC would still play a role? Is it really, it's about leaking behavior, or it's more generic, just like transfer of sensory into some motor action?
Yes, if it is possible to train mice to move their eye using very similar task paradigm, I think some studies talking about the mouse ACC is a little bit analogous brain area with the monkey FEF area. So I think it is possible that the ACC is also related with some orienting behavior, such as moving eye socket, but I didn't try that, so it is still unclear, I think. Yeah.
Okay. So then there is another question by Raunak Basu. Great talk. How does your model explain lack of leaking during the no-go signal?
Actually, the current behavior paradigm during the no-go trial, there is actually no stimuli, just catch trial. So it is there. In other words, there is no different type of stimuli during the no-go trial.
I see.
So yeah, so here the Raunak Basu told about the no-go signal, but actually there is the visual stimuli during the intertrial interval and the no-go trial. In other words, the catch trial is the same. So just they need to withhold their leaking action during the no-go trial.
So I think the question is that how would ACC implement that?
So I think during if there is no visual stimuli, in that case the mice have to withhold the leaking action, since there is no meaningful stimuli. In that case, the sensory increasing neuron have to be silent, and also other network activity has to be a little bit, they don't need to go up, and motor decreasing neuron is also don't need to go down. So all three key players, the number one is sensory increasing neuron, and number two is the motor decreasing neuron, and number three is the other network neuron, all cooperate together to withhold their leaking action during the intertrial interval and the background no-go trial.
Okay. Next question by Albert Koo. How much variability do you observe among the neurons during lick miss and perceptual categories, or the whole areas are highly coordinated? It's just about the heterogeneity of responses in ACC.
Yes. So when actually, as some maybe Albert Koo asked to me, so I already observed some variability across the different neurons I recorded in the ACC. So in particular, in case of the sensory increasing neuron, I collected total about the 60 ACC and sensory increasing neuron, but among them, about 20% of the sensory increasing neuron showed the significant correlation between the latency and the visually evoked signal.
So the rest 80% do other stuff.
Yeah, the other 80% did not show any correlation. And in case of motor decreasing neuron, I collected total 44 motor decreasing neuron. Among them, 20 motor decreasing neurons showed significant correlation between the lead response time and their neural activity. So maybe about half of them, showed the correlation, but there is some variability. Yeah.
Good. So I think we have time for one more question, and that would be question by Sachin Vanad. I'm sorry if I pronounced wrong your name. Have you looked at cross correlations between the spontaneous activity of sensory and motor neurons in the ACC?
Yes, that is actually the excellent question, I think. So when I calculated some cross correlation between the sensory increasing and motor decreasing neuron, I expected that some monosynaptic inhibitory connection from sensory increasing neuron to motor decreasing neuron. So among multiple pairs of the sensory increasing and motor decreasing neuron, I observed some possible monosynaptic inhibitory connection between that pair, but there's some, I think, some variation, not always reliable result. So yeah, I tried that, but some pairs show that, but the other pairs did not show that kind of correlation.
Okay, so I think we should stop here. Maybe one more, the last question.
Yes.
Victoria Corbett, what are inputs to ACC? What other inputs to ACC may cause the release of licking during spontaneous licking when there is no visual stimuli response?
Oh, yes. So I think ACC receive many neuromodulatory inputs, such as cholinergic neurons from the basal forebrain, and so some dopaminergic signals from dopaminergic centers. So maybe that kind of neuromodulatory signals can change the ACC neuronal activity to drive some spontaneous leaking behavior in the absence of visual cue.
Okay.
Yes.
Thank you, Jae-Hyun. So just to remind, you can create that session option, right? And then you can continue discussions with whoever wants to continue the Q&A with you after your talk. We have to move to the next talk now. Thank you so much.
Yes.
That was great.
Yeah, thank you for having me.
Thank you. Bye.
Bye.
Okay, so I think now we move to the second talk of today by Marion Rouault. Do we have Marion here?
Yes, can you hear me well?
Hello, Marion. Yes.
Hello.
Thank you so much. So Marion is going to present her work with Valentin Wyart from Ecole Normale Supérieure. And the talk is distinctive signatures of information seeking during changes of mind in controllable environments. Very much looking forward, Marion.
Thank you very much, Athena. Welcome, everyone. I'm very happy to have the chance today to present to you some of our work on information seeking during changes of mind. This is a line of work that has been developed in the team of Valentin Wyart in Paris, and that have involved quite a few people of the team. Humans can form and revise uncertain beliefs through information sampling, through sampling of new evidence, and seeking information indeed about the reliability of alternative strategies is essential for adaptive behavior. However, information seeking often co-occurs with changes of mind about the perceived reliability of the current strategy. What I mean here basically is that when you are seeking information, it is usually when you realize that what you are doing is no longer working, that you are not happy with the current strategy, and so it's a good time to seek information about alternative options. But this means then that the cognitive and neural basis of information seeking are usually confounded with those of changes of mind in general. So in this project, we have tried to isolate information seeking from changes of mind in general. And to do that, we leverage the fact that information seeking requires instrumental control. So to actually seek information, you need to be in a situation in which you have active control over the evidence you want to sample, over the information you want to sample. So to isolate these signatures of information seeking, we have contrasted the changes of mind occurring with and without control. So I'm going to explain how we have operated this notion experimentally. So in our experimental design, we had two generative categories, two generative sources, blue or orange, that lead to different possible oriented bars that you can see here at the bottom. So these formed our different stimuli. And on any given trial, that there was one of the two sources that was the latent source that generated a sequence of stimuli in the form of oriented bars. So each trial could have a sequence from 2 to 8 stimuli on which people could perform sequential Bayesian inference to try and estimate which was the latent source. So after seeing the sequence of samples, typically they'll have formed a belief about which is the most likely latent source. And the manipulation we have introduced is that we have created two conditions of controllability. In the first condition, the Q-based conditions, participants were presented with the stimuli, and they were asked to identify the sampled source. So they were asked to identify from which category the samples they've seen are formed. So they were asked to monitor the samples and report on what they think the category is. So in this condition, typically, participants are observers presented with external information about which they have no control, and they have to make a decision about it. So this is typically the case like in most perceptual decision-making paradigms. In contrast, in the outcome-based conditions, participants were asked to produce samples from either of the target source. So they were instructed to draw samples of either target category. So in these conditions, participants are in control of the evidence they see unfolding before their eyes. This is important because this is the key difference between the two conditions. In addition to response about the category, we also asked participants for a confidence estimate in their response, to respond high or low confidence for either of the two categories. Finally, we had reversal of the latent hidden source that occurs from time to time that required participants to constantly adapt, which is the current category at the moment. This allowed us, with this paradigm, to really compare the Q-based and outcome-based inference in tightly matched conditions that only differs by the degree of control offered to participants. We have looked at, first, the behavior of participants after a reversal. So we are looking here at the proportion of time that participants selected the true hidden category after a reversal. So you can see that after a reversal, participants learn and gradually adapt to select more often the true category. But importantly, we found using psychometric fits a smaller reversal time constant in the Q-based condition, meaning that participants were overall slower to reverse when they were in control of the evidence. We also found that they reached a slightly higher plateau performance in controllable environments, as illustrated by this higher asymptotic reversal rate. So in addition to choices, we have also looked at confidence after a reversal. So you can see that in both conditions, confidence decreased after a reversal, as you might expect, while participants are adapting to the reversal. And then the confidence gradually increased again. But importantly, we found a larger confidence drop in controllable environments, as illustrated again by psychometric parameters. So that's for choices and confidence. So we have looked at this data again, but this time we have replotted the data as a function of whether the evidence is consistent or inconsistent with my previous choice. So here we are looking at the proportion of time that the participants repeat their previous choice as a function of whether the evidence indeed encourages them to repeat their previous choice versus when the evidence conflicts with their previous choice. And what we found here in both conditions is that when the evidence in favor of repeating my previous choice increases, indeed participants repeated their choices more often. But there was a difference between the two conditions in the point of subjective equivalence here, so when you cross the horizontal line. And we found a larger point of subjective equivalence in the outcome-based condition, the pink one, indicating that participants needed more evidence to change their mind in controllable environments. So they needed more evidence against their current beliefs to be able to change their mind. And importantly, this was found in a context of a lack of difference in the sensitivity to evidence. So the slope was the same in the two conditions, indicating that participants were able to process the evidence equally well in both conditions. So it was not the case that they didn't pay attention or anything like that. We have looked similarly at confidence, the fraction of high confidence responses, again as a function of whether the evidence is consistent with my previous choice or conflicts with my previous choice. And so on the right part of the graph, when the evidence is consistent with your previous choice, this is where most repeat trials happen. And on this part of the graph on repeat trials, we found that more evidence leads to a higher confidence, as you might expect, in both conditions. However, when the evidence is inconsistent with my previous choice, this is where most changes of mind occur. We found that confidence was reduced as compared to repeat trials, and was reduced even more so in controllable environments in the outcome-based conditions. So we sought to explain a little bit the mechanisms behind these patterns of choices and confidence using a shared computational framework. And indeed, an important feature of our experimental design is that we were able to characterize the inference process identically between the two conditions. So I'm going to walk you through what the model does intuitively, hopefully. So first we had a stimulus presentation. So participants are presented with evidence samples drawn from these two categories, on which they have to make an inference. So we have inference noise that corrupts the way people process evidence, and that scales with the amount of samples presented. So that participants integrate into a posterior belief about what is the most likely category currently. So this is based on a normative Bayesian model, the most parsimonious way to solve the task. We have also a perceived hazard rate that models the reversal occurrence and corrupts the posterior. And then we have the response phase. So we have a very simple policy for decision here, by which decision and confidence are read out from the same posterior belief. So what we did is we took this model and fitted the parameters on top of for a participant's choice and confidence data. And I'm presenting here the best fitting model parameters in both conditions. So first, we found no difference in inference noise across conditions. Again, this is important because it means that participants were not more noisy in one condition or were not able to process the evidence equally well in some of the conditions. But importantly, we found a lower perceived hazard rate in the outcome-based condition, indicating that participants perceive the contingencies as being more stable in controllable environments. So they perceive the reversal as being less likely when they were in control. And so we took the best fitting model parameters to simulate the model as if it was a participant and plotted it here on top of participants' data. And this way we could find that with the simulations that the computational model is validated in the sense that it captures qualitative signatures of participants' choices and confidence. And then finally, in terms of behavior, we have looked at what happens after a change of mind. So we are looking here at the fraction of changes of mind that are confirmed on the next trial as compared to whether people go back to their previous response, as a function of whether the change of mind was performed with high confidence or with low confidence. And what we found here importantly is that changes of mind were more often confirmed in controllable environments, which we interpret as a form of hypothesis testing, meaning that in controllable environments, participants were more likely to test again their new hypothesis after a change of mind. And these behavioral differences were also visible in a number of neurophysiological signals. We have run the task in MEG, and we are looking here at a pairwise correlation matrix of the time courses of all of the MEG channels recorded. And when we look at this correlation, we can see that there are two distinct clusters that seems to appear, which we have called occipital cluster and frontal clusters, as well as a number of other channels. And in these two clusters, we were interested to look at the time courses of repeat minus switch trials, so repeat trials against changes of mind, in an alpha band power that has been associated in the literature with fluctuations in attention and arousal when you have this alpha suppression. And we've looked at the time courses time-locked at the response probe onset. So for these two clusters, for the repeat minus switch contrast, in the occipital cluster, we have first observed that the switch effect starts earlier and peaks before the response probe onset. Whereas in the frontal cluster, the switch effect starts a bit later and peaks after the response. So that's for the Q-based condition. In the outcome-based condition, we found an effect, this change of mind effect was stronger in controllable environments, with the strongest difference before the response probe onset. Whereas in the frontal cluster, again, changes of mind had a stronger effect on this alpha band in controllable environment, with the strongest difference occurring after the response probe onset. So that's for fluctuations in cortical signals. We have also looked at pupil dilation, which in the literature have been associated with fluctuations in the locus ceruleus noradrenergic system. Again, looking at the switch minus repeat contrast, time-locked from the response probe onset. And we can first observe that in both conditions, there was an increased pupil dilation on changes of mind trials as compared to repeat trials. And this increase was larger in controllable environments. Moreover, unlike MEG effect, we had a post-response effect that starts mainly after the response, and that is more extended in time, presumably still occurring during the decision evaluation phase. And so with that, I'd like to conclude. I hope that we have been able to isolate signatures of true information seeking, independently of changes of mind. We observed in controllable environments that changes of mind required more evidence against current beliefs. In controllable environments, changes of mind were associated with reduced confidence, but were also more likely to be confirmed on the next decision. And information seeking was associated with neurophysiological markers of increased attention and arousal, in particular, pre-response visible in MEG in the alpha band, and in pupil dilation post-response markers. So we have a preprint with a bit more information about the model. And I would like to finish by thanking the organizers of Cosyne, and thank you very much for your attention.
Thank you so much, Marion. That was excellent. So please, everyone, share and put your questions in the Slido. Maybe I start with one question, which is about these changes of mind that require more evidence in a controllable environment. So you modeled it through this kind of a hazard rate
Yes.
parameter. So I was just wondering, how does that really interact with the prior? Because I would just imagine that like priors of participants in the controllable environment is stronger toward the current belief, right? It can be just a kind of if you just simply model it with a Gaussian distribution, it's just the narrower, low variance prior compared to the other condition. So, but I'm not sure whether you modeled it this way.
So you are absolutely right that the finding of a lower perceived hazard rate in controllable environments is associated with stronger prior belief. So basically what the hazard rate is going to do is going to squish your beliefs closer together. And you are right that in controllable environments, it means that people will have a stronger belief either way in favor of either category. So this is related. And the consequence of that is that then to be able to change their mind, they need more evidence against these current beliefs to be able to shift their belief about the category when they are in controllable environments.
Interesting. And how was the instruction for participants? Did you explicitly told them that they have more control in this environment, or they really need to also infer that, learn that condition and that environment?
Indeed, we wanted not to instruct them explicitly. So there was a game. It was meant as a game of cards. So they had either to report on which deck the card had been drawn, or they had to click to produce cards of either deck. So the notion of control was not introduced explicitly here. It's through the experience of the task.
There is a question. I think maybe we really need to address that. Somebody asked the question, Gautam Agarwal, and it received a lot of votes. So I didn't understand the controllable version of the task. Would you mind explaining it again? So maybe you should kind of, tell us a little bit about that.
Of course, no, this is the key difference here. So in the controllable condition, participants were presented with a target category, and they had to press either of two keys to produce the category. So they had to figure out a mapping between what motor key produces what category. So it means that in this condition, the evidence that they see is the consequence of what they've pressed, of what they have produced. So this is what we mean by controllable here. Maybe it's not the only word possible to explain this difference. An alternative is to think of the outcome-based condition as a condition of active sampling versus the Q-based condition as a condition of passive sampling. So in the Q-based conditions, people see the evidence. They are external observers, and they have to report about what they see. Whereas in the outcome-based condition, people are instructed to produce stimuli, to draw stimuli. So that is how we operate this notion of control. I hope this helps.
And they could go back and forth between the two categories just as well. So they could just like press whatever key they wanted in order to produce samples.
Absolutely. So they had to. So they had an instruction about which category to produce for this current trial. But you are completely right that it is through the keys they press that they can really test their hypothesis.
Okay. So then let's move on to next questions. How is the hazard rate calculated? And somehow the model modulates the prior described in this model.
So the hazard rate was fitted for each individual participant and for each condition on the basis of participants' choices. This does not depend on confidence, just on choices. So it's, really via a fitting procedure that we established that in the outcome-based condition, on average, participants had a lower perceived hazard rate.
Okay. There is another question by Tyler Meredith. So as I understand it, you use the model to estimate their perception of the hazard rate. Did you also ask them what they thought the hazard rate was?
That's a very good question. So no, in this version of experiment, we have not. We have an alternative experiment in which people were cued about the volatility. So they were blocks of trials in which they expected reversal to be more frequent or less frequent. We have that in the preprint, which changes a bit their perception of the volatility. But here we didn't ask for explicit reports. We don't have this data.
Okay. Maybe last question, because we are almost over time, by Jeff Ehrlich. I also didn't understand whether the reversal was with a trial or across trials. And I think then he had a follow-up question, and was a change of mind within a trial or across trials?
So the reversal occurs across trials, across sequences, as you would have in typical reversal learning paradigms. Sorry, what was the second part of the question? The changes of mind. No, the changes of mind we look from trial to trial. So basically, a change of mind is when you change your response as compared to the previous choice. The opposite is a repeat trial in which you repeat the same category.
Okay, so maybe really the very last question. Was the alpha range evaluated for each participant or arbitrary?
Sorry, could you repeat that?
Was the alpha range evaluated for each participant or arbitrary? I'm not quite sure whether I understand that, what they mean by arbitrary. I'm wondering because the upper limit, 18 Hertz, seems to overlap with beta range commonly defined.
So this is a question about
I see.
I think it was the same window range. I need to double check that, the same window range across participants. If that's the question, sorry.
Yeah. Thank you so much, Marion. That was excellent. Thank you so much for the talk and discussion. Bye.
Thank you, bye.
Okay, now we move to the last talk by Danique Jeurissen and Löffler. Let's have them on the stage. I think we only have Danique at the first. Hi, Danique.
Hi.
So Danique is from the Mike Shadlen lab, and she's going to present a work which is joined by Anne Löffler from Daniel Wolpert and Mike Shadlen lab. And the talk is the title of the talk is Serial Time-Multiplexed Incorporation of Evidence to Make Two Decisions About One Object. We're looking forward, Danique. Thank you.
Thank you so much, Athena. It's so much fun to be at Cosyne this year, and I look forward to the in-person ones in the next years. So can you see my slides and hear me well?
Yes.
Okay, great, thank you. Okay, so today I'll be presenting a project that came out of a collaboration between the Zylberberg, Wolpert, and Shadlen labs. And Anne is the person who collected the data with the human subjects, and she'll be joining us for the Q&A session today. Okay, so imagine we're on a hike together, and suddenly an animal appears. If it's a lion walking towards the right, we may want to hide behind that big tree on the left. But if it's a deer, we may not be in immediate danger. So in other words, it's not just the direction of the animal's movement that will determine our behavior, but we will also want to look at the animal's color to determine what we should do. And as speed is of the essence, we would ideally make these two decisions simultaneously. So in this example, the decision about the direction of motion is independent of our decision about color. And to make a decision about the conjunction of these two features, the lion is walking towards the right, we need to combine the evidence along two feature dimensions. And today we'll talk about how these two decisions can be combined. So I live in a place where I do not encounter any lions, but what I do see daily are these moving dots. But we can still ask the same question here about the direction of motion and the dominant color. And that is exactly what we did in the lab to answer the question, how do we make multiple decisions about motion and color? We use a reaction time version of a random dot motion task. And subjects report a conjunction of motion and color in a four-way choice. They choose left and blue, left and yellow, right and blue, or right and yellow. And human participants hold a handle that they move to indicate their choice. We use a robotic interface to constrain the movement to the relevant diagonal paths. Now, if our video connection is working well, you should see that in this example, the dots are moving towards the right and are a majority blue. Subjects indicate a correct answer by moving the handle to the right blue target. And the participant is in control of the stimulus duration. The dots will disappear once the movement is initiated, and we measure both choice and reaction time. Importantly, the subjects need to complete both decisions before they can indicate the answer. Across trials, the difficulty of motion and color varies independently. And we collected data from 8 participants in this experiment. They each completed at least 2,000 trials. Now here we're plotting the proportion of rightward choices on the y-axis and the motion strength on the x-axis. When motion is strong towards the right, subjects correctly report a rightward choice. When it is towards the left, they report a leftward choice, with intermediate levels in between. Now, when we plot the proportion of blue choices as a function of color strength, we again see a very good choice accuracy. Now, both of these plots only show the data of trials in which the other dimension is easy. But interestingly, when we overlay the choices of the other difficulty levels, we see similar choice functions. The overlap of these choice functions means that there is no interference in choice accuracy. In other words, the difficulty of one dimension does not affect the accuracy in the other dimension. Now, the absence of interference in choice can be explained in two different ways. First, given the parallel processing of motion and color in the visual system, these decisions might evolve in parallel as well, with each dimension using a separate processing stream. Alternatively, the two streams of evidence might not be able to influence the final choice simultaneously, and instead the two decisions might evolve in a serial manner, using separate time epochs. We cannot distinguish between these two options by looking only at the choice functions. So instead, we will need to look at the reaction times as well. And in previous work, drift diffusion models have been used to explain the observed pattern of choices and reaction times. Today, we'll consider two versions of such a model, a parallel model and a serial model. Now, in this example, we'll consider a stimulus with low coherence rightward motion and the clear majority yellow dots. This gives us two streams of momentary evidence. Motion will be visualized at the top and color at the bottom. Now, across time, momentary evidence is summed to generate a representation of the accumulated evidence. And here you see that the difficult, low coherence motion information slowly accumulates towards the upper bound, indicating a rightward choice. At the same time, you see that the easy color information quickly accumulates towards the lower bound, indicating a yellow choice. Evidence for each decision is accumulated independently, and each is associated with its own decision time. T motion is quite late because that was the harder decision, and T color is much earlier. Now remember that participants only report their final choice about the conjunction of the two features. I'll call the time when the combined choice is reported the double decision reaction time. If the two choices are made in parallel, the double decision reaction time depends on the maximum of T motion and T color. And this parallel process is a good candidate. We know that there are specialized neurons in the brain that code for either motion or color, and integration of the two streams of evidence simultaneously would be the most efficient way to solve the task. However, maybe it's not possible for the brain to integrate two streams of evidence simultaneously, and instead, we may use a serial strategy to solve this task. Such a strategy could indicate that the two decision variables cannot be updated simultaneously. In this example, I'm showing a trial in which accumulation of motion evidence is completed first, which is then followed by the accumulation of color evidence. The choice would remain the same, right and yellow. But now the reaction time depends on the sum of T motion and T color. To prepare you for the pattern of reaction times I'll be showing you soon, I would first like to give you an intuition of how the models differ in their prediction. First, let's select only the trials in which color is easy, and we expect that when we sort the data now by motion coherence here on the x-axis, that subjects are faster if motion is easy, slower when motion is difficult, with intermediate reaction times in between. And many of you might recognize this as a similar pattern of reaction times we often see when making a single decision. And indeed, it is very similar, because color is easy and does not add much time to the total reaction time. The crucial comparison is now to the trials in which color is difficult. Here we expect that the reaction times slow down with a delay that is constant across all motion coherences. The time added for trials in which motion is easy is the same amount of time as added for trials in which motion is hard. The predicted reaction time is simply the sum of the time needed for color and the time needed for motion. In other words, the graphs will look like stacked versions of the same curve, because increasing the color difficulty just adds additional processing time for color without affecting the time needed to decide on motion. Now, in contrast, the parallel model makes a different prediction for the pattern of reaction times. When color is easy, reaction times still depend on the difficulty of motion. However, the parallel model predicts that when color is difficult, that this mostly affects the reaction times for trials in which motion is easy, and that reaction times are only slightly increased when motion is hard. In other words, the reaction times resemble the time needed to complete the most difficult of the two decisions. If one of the two dimensions is easy, it shares the time with the other, more difficult decision, and it will not cause much of a delay in the total reaction time. When both decisions are hard, either one of the two decisions could be the slowest one, and this is the one that determines the total reaction time. The graphs will therefore look as if the curves are squeezed together in the middle. Okay, so next we'll look at the double decision reaction time of the human participants. On the left, we plot the reaction time as a function of motion strength, separately for each level of color difficulty. On the right, we plot the reaction time of the same trials, but now as a function of color strength, separately for each level of motion difficulty. When we plot the reaction times for the easy condition of the other dimension, here in blue and purple, we see a dependency on stimulus coherence. When we plot the reaction times for the hard condition of the other dimension, here in green and red, we now see reaction times with an additional offset. And for intermediate levels of difficulty, we see intermediate offsets. I hope you're already starting to recognize the pattern of reaction times of the serial model. And indeed, when we add the predictions of the serial model, this closely resembles the data. For comparison, let's now take the exact same data points and overlay the predictions of the parallel model. This model is not a good fit, and it especially fails to predict the long reaction times that we see when both color and motion are difficult. So from the double decision reaction times, we conclude that double decisions are made in a serial manner, using separate time epochs. In other words, we saw before that there is no interference in choices, and the reason for this is that these decisions are not made at the same time. But we don't fully understand yet how the total time is allocated between the two decisions. So to understand how the time is allocated to each of the two decisions, we will consider a single switch and a multi-switch model. We already discussed this single switch model, in which the evidence for one decision is accumulated first, followed by the accumulation process of the second decision. But alternatively, we may be switching back and forth between the two decisions, first accumulating a little bit of motion evidence, and then, while holding on to the accumulated motion information, we would accumulate evidence for the color decision, and then switching back and forth between the two until one finishes. From then on, the remaining time would be devoted to the unfinished dimension. And regardless of whether you finish one decision and then the other, or you alternate back and forth, the prediction of the reaction times remains the same. It is still the sum of t color and t motion. To distinguish between the two possibilities, we need to know when the first decision finishes. But with the unimanual version of the task, we do not have access to that information. So we therefore designed a bimanual version of the task. So to get access to the information about when this first decision finishes, we asked the participants to perform the double decision task again, but now using two hands. In this example, the decision about color will be indicating by moving the left arm up or down, while the decision about motion will be indicating by moving the other arm towards the left or right. Participants are encouraged to report each of the decisions whenever they are ready. The stimulus doesn't disappear from the screen until the second response is initiated. And in addition to giving us information about when the first decision finishes, this experiment has an additional advantage. It allows us to test whether two decisions can be made in parallel if the motor outputs are independent. Participants might then be able to keep the two streams of visual information as well as the motor plan separate, which might be the optimal condition to allow for parallel processing. But consistent with the first experiment, we find that subjects still process the two decisions in a serial way. So similar to the type of data we looked at for the unimanual task, we will first take a look at the time point at which both decisions are completed. So data is plotted in the same format as before, but now we look at the reaction time of when the second arm moves. So based on the fits of the unimanual data, we can now generate predictions for the double decision reaction times based on the serial model, represented by the lines here. When we now overlay the data from the new bimanual task, we see that there is an excellent fit, meaning that participants still use the same strategy of serial integration. When we take the same data points again and compare it to the parallel model predictions, we see a poor fit to the data. Now, this result was somewhat surprising to us. Even when the circumstances are optimal for parallel processing, we see that the two streams of visual information and the two motor outputs are separate. the decisions are still processed in a serial way. So our task may have revealed a fundamental limitation in processing multiple decisions that is not due to generating the motor response. So now that we've examined the time that it takes to complete both decisions, we can now look at the first decision to address the question of whether subjects complete one decision and then the other, or whether they switch back and forth. So first of all, the models predict that the same double decision time will happen at the end of the trial, which indeed we've already confirmed for the bimanual dataset. The single switch model predicts that the time it takes to report the first choice, which I will call RT1, does not depend on the difficulty of the second choice. The multi-switch model predicts that RT1 is modulated by the difficulty of the other choice. And to fit these two models, we added two parameters. The first one is the probability of starting the process with integrating the motion information. The second parameter is the frequency of switching between the two dimensions. So let's compare the predictions in more detail. We will split the data by whether motion was reported first or color was reported first. And on the y-axis, I will plot the response time of the decision that was reported first. On the left as a function of motion strength, on the right as a function of color strength. The single switch model predicts that if motion is reported first, the response time will depend on the motion strength. And if we plot the same data as a function of color strength, we would see no dependence on color coherence. Now, when color is reported first, we would see that there is the reverse pattern. RT1 would not depend on motion strength, but would depend on color strength. In contrast, in the multi-switch model, RT1 would depend on both the motion and the color strength because they share the time. And we would see a similar pattern when color is reported first. The data closely matches the blue lines, meaning we see strong support for the multi-switch model. This suggests that participants switch back and forth between the two decisions. So from the study I presented today, we conclude that multiple decisions about one object are made in a serial manner. Next, double decisions are serial even when motor outputs are independent. So we think that the seriality is due to the fact that the two decision variables cannot be updated simultaneously. And from looking at when the first decision finishes in the bimanual task, we conclude that participants switch back and forth between the two decisions. So thinking back of the example where we encounter a lion in the forest, we cannot decide on the animal's direction of motion and its color at the same time. The two decisions compete with each other, which leads to additional processing time. And we think that this slow processing might arise from a need to flexibly use the same neural circuitry for different decisions. And to better understand the neural circuits, we are currently recording in parietal cortex of monkeys performing the double decision task. So last, I would like to thank everyone in the Shadlen, Wolpert, and Zylberberg labs. It has been a real pleasure to work with everyone on this. And most importantly, I would like to thank Anne and Yul. And Anne will be joining us for the Q&A now. So we'll be happy to take any questions. With any remaining questions, please also take a look at our bioRxiv paper, and you can tweet at us.
Thank you so much, Danique. This was excellent. There are so many questions. So I'm actually going to suppress my own question and just start going through the questions. If you can have Anne on stage, that would be great. Okay, the first question by Jan Drugowitsch. He's asking, do you think that your results might change if you use two different modalities, for instance, visual and auditory, instead of two visual features?
Anne, do you want to take the first?
First of all, hi everyone. It's good to be here, and I'm happy to take that question. That's a really interesting question. So we haven't tested any auditory and visual modalities yet. However, we do believe that, so we do have some evidence to show that this bottleneck does not operate at the level of the sensory input. We actually show that the sensory acquisition itself can happen in parallel, and that the serial processing really happens at the stage of the decision variable that is being integrated. So we do think that this is sort of modality independent, and probably if we had a decision-making task where you over time had to integrate auditory information, you would still see this seriality of the process, probably.
Do you mind if you can kind of elaborate a little bit how you disentangle the sensory processing from decision?
Right, so we have one experiment where we only control the viewing duration of the stimulus. And in one condition, we only show the stimulus for a very short amount of time. and we are performance in this double decision task to a task where they only judge either the motion or color dimension. And we show that in this case, there's no interference in performance still, even though you might expect if you only have a short amount of time, if you had to switch between them, you would miss out on information on either stream, but that's not actually the case. So that shows that both streams of evidence, the color and the motion, are acquired in parallel at the start of the process, at least for short snippets of time.
Okay, I have a lot of questions, but maybe we should take them offline. So I go through the rest of the questions posted here. Next one is by Kenway Louis. Are there intermediate possibilities between serial and parallel? Maybe an attention-based one, where the end of one drift allows a stronger gain on the other one.
Yeah, so we considered several versions and mixes of the model. For this talk, we decided to only sort of put the two most extreme versions out there because we find such strong support. But there are different ways to look at this. So there are target-wise or mixture models. The strongest support that we find is for this complete serial model, though, and I'm happy to go into more detail of the how the fitting procedures work and how we find that support. But related to the part of the question to attention, so we do see some parallels to the attentional literature, of course, of switching back and forth, maybe having feature-based attention between color and motion. So yeah, I don't know how that would change the model exactly. I would have to think about the drift at the end for how it changed the gain of the other. So maybe I can talk to this person in more detail to understand how that would work exactly. But there are a lot of parallels to the attentional literature here as well, I think.
Okay, so yeah, just to remind again, you can create this session and then continue discussion with people who are participating in the Q&A offline. Okay. The next question is by Sophie Deneuve. She's asking, did you try a more optimal model, which would be summing the log odds ratio of the two choices and then make a decision when some of them reach a threshold?
So I think that question is similar to what Danique just talked about with a target-wise integration. And that model would actually predict interference on the choice level between the two dimensions. So we can rule out this group of models simply by showing that there's no interference at the level of choices, but it's really just in the reaction times where we see our model confirmed, basically. Yeah.
Next question by Lena Salfen-Moser. So were the participants asked how they made their decisions? Were they asked whether they did parallel or serial decision?
No, we didn't ask participants. We didn't ask them, and from own experience, when you do the task, you might feel like you do it in parallel, but every participant we tested so far shows very strong support for serial processing.
And so do you think it's possible to kind of bias the subjects in any way? Like if you really want to do parallel, can you?
So one thing I can say about that is that if a lot of training would make people parallel, if there is a way to sort of get around this sort of bottleneck of processing one after the other, then we will probably see that after a lot of training, which is exactly what we do with the monkeys. And even after a long training process, they still seem to show this serial processing. So we don't seem to be able to get rid of this bottleneck anytime soon. Yeah.
Next question by Claudia Kloppert. She's asking, do you think that the switch frequency depends on the difficulty of the task or some other task parameters? What determines the switching frequency?
Right, so by default, in a way, so when both dimensions are hard, that's when you get the most switches, just because you have to spend more time on both dimensions and you have to continue switching forth and back until one of them finishes. But it is an interesting question in general. How exactly is that switching frequency determined? Are there any top-down processes? How do we select a dimension that we start with, and so on? So I think there are a lot of interesting questions that are still kind of open.
Okay, so maybe just one or two more questions. Next one is by Tatiana Engel. She's asking, how are your results related to Rangel's work on value-based decisions, showing that evidence is only accumulated while a subject is attending to the object?
Anne, do you want to take this one?
I'm not sure what the relation is to value-based decisions, but as we mentioned before, I think it probably is related in a sense to maybe attention, and that you're kind of switching forth and back between the two features of that one object that we use here, at least at the level of the decision variable. But I'm not sure how that's related to value-based decisions, and I don't know if there have been tasks in that literature where you really had two features where you had to integrate information over time.
Okay, so we are a little bit over time, so I ask one last question, but please continue the discussion offline. So let's see, the next highest voted by Ramon Nogueira. So have you considered asking subjects to perform XOR or other combination tasks between color and direction?
Yes, I think if you would do only, like, even if there are two dimensions in the stimulus, but you would only have to use one of the dimensions, so it clearly or closely relates to the task that Valérian Monte did a few years ago. So people are well able to suppress just one dimension and only decide on the other. In addition, we've also done a task, and can talk about this more as well, where you have to look at two patches of motion. We see serial processes there as well. So this does seem to come back across all kinds of different tasks that we do. And maybe just to add to that, one of the things that we can do then in the monkey and look in the electrophysiology is really see, like, how is this information coming in, and how are these decision variables maybe sort of kept separate in areas like LIP, where we can start to see, are there neurons involved in the motion decision, neurons that are involved in the color decision? Do we see alternations? Can we sort of like trigger at one group of neurons is starting to integrate and see what the other group is doing? So those are sort of the questions that we're thinking about for the future as well.
Okay, thank you so much. I have to stop here. We are kind of over time, 5 minutes. I mean, overall, like 14 minutes over the session, but it was excellent. Please continue, create this session and continue the discussion offline. Thanks a lot, Danique and Anne. Thanks a lot, everybody who were with us. And please join us at 11:40 Eastern Time for the next session. Bye.